All right, guys, welcome to Unit 6, our final unit of the year. We're going to be doing this one a little bit differently, obviously. I haven't given you bill notes, and the reason I haven't given you bill notes is because you're going to be working on the guided tutorials as we go through this unit, and those are going to sort of count as your bill notes. I think they'll be helpful to you in referring back to the information that we cover in this unit. But if you at any point feel the need to take bill notes, if it would just make you feel better to sort of keep going the way we've been doing previously in the year, feel free to take notes on these PowerPoints. I'll walk you through it a little bit more in detail during this one, and then in the subunits to follow, I won't explain it as detailed. But so this is Unit 6, Gene Expression and Regulation. And I wanted to give you this little list, and I'll post this PowerPoint so that you can see it on your own. But these are helpful videos that will help you with each of the sections of this unit. Most of them are Bozeman. There are a couple crash course videos, a TED Talks, but feel free to refer back to those at any point. The overview, there are 8 subtopics in this unit, and we're going to go through it in about 5 days or 6 days. So it's going to be fast, but we can do it. I'm going to start off with a short free response question that I want you guys to do on your own. When I tell you to, you can pause the video and come up with a quick answer, and then I'll walk you through it on the next page. In the course and exam description for AP Bio this year, they gave suggested skills for each unit, and I think these are really going to come in handy as the exam has changed and it's only free response questions. So I figured I would incorporate a couple free response questions in here so we can get some review in as we go through this last unit since we're short on time. So the suggested skill for 6.1 is to explain biological concepts, processes, and or models in applied context. So when they say applied context, they mean they're going to give you a scenario. It's probably going to be a graph or a diagram or a table, and you will have to analyze it and explain concepts that are being shown in this diagram. So in this example here, my suggestion on this type of question is to first, before you even read the question, figure out what the diagram, graph, or table is telling you. Basically, what's the story behind this that's going to help you answer your question without any context from the question itself. So if we were to look at this, always look at the title. So this is messenger RNA expression levels. So the expression of messenger RNA in different tissue types for different genes. So each tissue, it's looking at different genes within that tissue and which genes are being expressed and which genes are not being expressed. So the white boxes represent no mRNA from those genes. So in the liver tissue, these 3 genes are being expressed in high amounts, and these 3 genes are not being expressed at all. So we know liver tissue usually deals with detoxification, so maybe that'll come in handy. Then heart tissue, this gene is expressed in high amounts, and these 2 in moderate amounts, and then these not at all. Brain tissue, and you can go on and see. So what I want you to do is analyze this. Notice which boxes are notice if you can see any patterns, anything at all. And then I want you to answer part A. Based on the data provided, identify the gene that is most likely to encode a protein that is an essential component of glycolysis and provide reasoning to support your identification. So pause the video now, take about 4 minutes to answer this question, and then play back for the answer. Okay, so let's go on to the scoring guidelines. This is going to be similar to part B of question 1 on your free response exam this year. On your AP exam this year. Part B is going to ask you to identify and justify experimental design procedures. So part A asks, based on the data provided, identify the gene. So it's asking you for one of the 6 genes. Identify the gene that is most likely to encode a protein that is an essential component of glycolysis. Well, we know that glycolysis is important for all cells. It's how cells get energy. So all cells must perform glycolysis. So if you look at it, which gene, it's asking you to identify. The action verb is identify. It's a 1-point question. If it asks you to identify, look at all your options. So there are 6 gene options. You are asked to, after carefully considering all the options, choose the one that best fits the phenomenon or situation. This should be 1 sentence in length. And if you look back here, the only gene of the 6 that's found in all tissue types is gene G. All of these tissue types must perform glycolysis in order for their cells to survive. So gene G must be responsible for glycolysis. Don't just write gene G though, right? Gene G is responsible for glycolysis. Then it also asks you to provide reasoning. There are 2 ways to say this. You could say justify or you could say provide reasoning. Here they say provide reasoning. When you are asked to provide reasoning, you take the data and or content knowledge and use that to provide accompanying evidence to support a claim or statement. So in your claim, evidence, reasoning, this is like your evidence. You take the data and support the claim. It's usually 1 or 2 sentences. Excuse me. And like we said before, gene G is the only gene expressed in all 6 tissues, and therefore, because glycolysis occurs in all 6 tissues, it must be gene G that's used to perform glycolysis, okay? So hopefully that helps. We'll do a couple more of these as we go through this PowerPoint, not today. All right, so before you continue on, I want you to pause again, and I want you to either click this link in the PowerPoint, or I want you to just Google PBS, The Gene, Part 1, Dawn of the Modern Age of Genetics. And it's going to bring you to this show on PBS. Sorry, hold on one second. Anyway, so just Google that, and I want you to watch this documentary from minute 44 to minute 60. It's 15 minutes. It's going to give you a great introduction into Unit 6. The whole documentary is so, so good. I think it's 2-hour clips, and if you're interested in something, I recommend watching it. It's a good review as it is, but these 15 minutes are really good at introducing DNA and everything we're going to talk about. So please watch that now and then come back and continue this PowerPoint. All right, so the way we are going to go through this is I'm going to give you a very brief introduction to each section of the PowerPoint or of the unit, and then I'm going to stop and allow you to work at your own pace as you go through the guided tutorials, take the quizzes in the guided tutorials. They might feel like a lot of work and they might feel annoying, but I think it's really honestly the best way for you to work at your own pace to really learn this material. I think me talking to you for 5 days on the stuff isn't going to do much for you. I think if you actually have to take the quizzes, and as you take the quizzes, you'll see if you get a question wrong, it reappears. You can't finish the quiz without getting them all correct. So you really have to learn this stuff. So since we're in crunch time, that's the plan. So College Board gives us, as I said, the course and exam description for AP Bio. And the way they set up each section, I think I've showed you this before. 6.1, the title of 6.1 is DNA and RNA structure. And this should be a review for you at this point. They'll always give us an enduring understanding. So this is what we should understand at the end of this section. And the enduring understanding for the first several sections of this unit are that heritable information provides for continuity of life. So basically, we are all connected through DNA. All life on Earth contains the same genetic code. We are all connected. This suggests unity among all living things. descent with modifications, so we're all a little bit different, but we've all descended from one common ancestor. And the learning objective for this section is to describe the structures involved in passing hereditary information from one generation to the next. So that hereditary information is DNA and RNA. So that's our learning objective. We want to be able to explain that. And the essential knowledge that we need to gain as we do that is that DNA, and in some cases RNA, is the primary source of heritable information. For our purposes, it's almost always DNA, but there are a couple rare cases where RNA is the primary source. Genetic information is transmitted from one generation to the next through DNA and RNA. And genetic information is stored in and passed to subsequent generations through DNA molecules, and as we said, in some cases, RNA molecules. And then we're going to talk a little bit specifically about prokaryotic organisms, which have circular chromosomes, and eukaryotic organisms, which have multiple linear chromosomes. And then prokaryotes and eukaryotes can contain plasmids, which are small extrachromosomal double-stranded circular DNA molecules. So those are the essential knowledge pieces that we're going to gain as we go through this PowerPoint. Before you start, I know I keep stopping you, but I want you to do biozones 92, 93, 94, and 95 before continuing on with the presentation, because I think they'll give you more background knowledge that will explain what we're talking about a little more. So pause here, do those biozones, and when you're finished, come back. All right, so the second learning objective, is this the second one or did we do this? Yeah, the second one is to describe how the characteristics of DNA that allow it to be used as the hereditary material. So, what is the structure of DNA? DNA and RNA both exhibit nucleotide base pairing that's conserved through evolution. We've talked about this before. Adenine, that we represent with an A, pairs with thymine, that we represent with a T. Or uracil, in the case of RNA, also pairs with adenine. And then cytosine pairs with guanine. Okay, so this is Chargaff's rule that A pairs with T and C pairs with G. We're also going to talk a little bit about how purines are one of the categories of nucleotide bases. The purines are G and A, and they have a double ring structure, and the pyrimidines are C, T, and U, and they have a single ring structure. So if you look here, you can pause and read through all of this, but basically, remember there's a sugar phosphate backbone of DNA. So the backbone consists of a phosphate group bound to a sugar, which is connected to a base, bound to a phosphate, to a sugar, to a phosphate, to a sugar, to a phosphate. If it's single-stranded, then there's only one of these. If it's double-stranded, they run anti-parallel and form this double helix structure. And here you can see the purines, adenine and guanine, that have the double ring, and the pyrimidines, thymine and cytosine, that have the single ring structure. Uracil, if we were talking about RNA, is also a pyrimidine. And then these DNA strands are held together by weak hydrogen bonds. A and T are held together with 2 hydrogen bonds, and G and C are held together with 3 hydrogen bonds, just due to the nature of their chemical structure. And then this is actually more for the next unit, but we'll talk about, this is called the central dogma, where we go from DNA to RNA to protein, but more on that later. There's a scrollable interactive, if you're interested, that sort of shows you how these pair together. Feel free to practice that. This is just showing you the difference between DNA on this side and RNA on this side. And you can see uracil and thymine are almost identical in their chemical structure, except for this small CH3 that's missing on the uracil, but that's why they can be interchanged so easily in DNA and RNA. And then I just want to briefly go over the different types of RNA here. DNA is really just the double helix that stores our genetic information. RNA is a lot more useful, I guess you could say. DNA is important because it contains the information, but RNA is actually how we get that information into a protein. So it can form a lot of different structures. So We've got messenger RNA, which is essentially just the direct copy of the DNA code. It's one single line. It's a single strand of nucleotide bases with the sugar phosphate backbone, and it's read by the ribosome and produces a protein, which we'll talk about. Transfer RNA, it's still that single strand, but if you follow that single strand in a line, it sort of loops back in and bonds with itself and forms this weird little like cross-shaped structure. And the transfer RNA is important when we talk about translation. And then the ribosomal RNA almost functions more like a protein. It's still that single-stranded RNA molecule, but it folds back up into itself in this really intense, complicated structure that helps to form a ribosome that will read the messenger RNA and take the transfer RNA from, take the amino acids with the transfer RNA and build this protein together. So if you're interested, Biozone 98 talks a little bit more about the different types of RNA, and we will put it all together more in Unit 6.4. So at this point, stop this PowerPoint. You are going to continue on with the plans that are on the portal, and we will start 6.2, I believe, tomorrow. Okay. All right. Thanks. Hope that was helpful.